This recording is the nervous system and nervous tissue, part 1 of 2. The nervous system can be broken down into two main divisions. The central nervous system is the center of the nervous system and includes the brain and the spinal cord. This part of the nervous system is responsible for integrating or processing sensory information as well as initiating motor responses. It includes neurons that carry information down to areas of the body as well as receiving information coming from areas of the body. If information is moving up towards the brain, we refer to that as ascending or afferent information. If information is going from the central nervous system out to the periphery of the body, we refer to that as descending or efferent with an E information. There are ascending and descending or afferent and efferent neurons. The peripheral nervous system is outside the central nervous system, and this includes cranial nerves and spinal nerves, which help to get information to and from the central nervous system. Again, this includes afferent and efferent or ascending and descending neurons. Basic functions of the nervous system and nervous tissue can be broken down into sensation, where there is some kind of input or stimulus, integration, where that sensory input is processed, and then a response, which occurs as motor output. Usually, motor output goes to muscles or glands, which are known as effectors, which is why on this particular slide you will see the word effector in front of the word cells on the lower left-hand side of the screen. Basically, sensory information comes in through the peripheral nervous system, is integrated in the central nervous system, and any output that is sent in response goes out through the peripheral nervous system. Sensation is the process of receiving information about the environment to gain input. When we are aware of input or some kind of stimulus, that becomes conscious perception. Sensation gives us information about what is happening externally or outside the body, as well as internally or inside the body. One example of sensation is what we can sense by special senses, including things like taste and smell, where the stimuli include chemical substances. Touch is another form of stimulus that can be triggered by physical or mechanical changes. Sight is another type of sensory stimuli that occurs with light on photoreceptors, and hearing is another type of stimulus that involves sound waves. Sensory stimuli from inside the body could include different things like stretch receptors in the walls of organs, or muscles or tendons, or from the concentration of different ions in the bloodstream. Here we have another example of sensation, integration, and response, where the received stimuli from the eyes, this is visual input, goes to the central nervous system to be processed by that portion of the nervous system, and it is at this point in the central nervous system that the stimuli will be compared with or integrated with other stimuli that are occurring at the same time, past stimuli that the individual has experienced, or even the state of the individual at that particular time. From that point, specific responses will be generated. I'll give you an example of this. If a person sees a glass of water and his brain processes the fact that there is a glass of water, if the person is not hungry, the motor output would be to walk away from the glass of water. If the person is thirsty, then the motor response would be to pick up the glass of water and drink it. Again, integration depends on a lot of different things. A response is going to be produced based on the stimuli received by the sensory structures after perception has been processed by the CNS. The difference between sensation and perception, a sensation is merely the reception of some kind of stimulus. The perception occurs when the central nervous system processes the information. One individual might pick up a cell phone and perceive it to be very small. Another person might pick up the same cell phone and perceive it to be bulky, because perception is how the brain determines what the sensation represents. There are two forms of responses that can occur in the nervous system. One is a voluntary response. We've already seen voluntary responses occur with the contraction of skeletal muscle. The voluntary portion of the nervous system is referred to as the somatic nervous system because it does require the initiation of thought in order for that portion of the nervous system to be activated. The other type of response that can be generated is generated as an involuntary response through a different portion of the nervous system referred to as the autonomic nervous system. Whenever I hear the words autonomic nervous system, my brain automatically translates that into something that's automatic. We do not have to think for the autonomic nervous system to function, which is really good. The autonomic nervous system includes contraction of smooth muscle, cardiac muscle, and glands. The somatic nervous system is going to involve conscious perception of what's going on in our environment. Imagine a person riding a bike and all of a sudden there's a hole in the path of the bike. That person has to use his or her senses to physically maneuver the bike so that the bike doesn't go into the hole, causing the individual to have a crash. The somatic nervous system not only involves conscious perception, it also includes voluntary motor responses in the form of contraction of skeletal muscles. This includes reflexes that happen without a conscious decision, as well as motor responses that become automatic as a person learns motor skills. When we walk, we're not constantly thinking right foot, left foot, right foot, left foot. Our brain knows where we want to go, and then we move in that direction. The skills that we learn become a function of a pattern that has been facilitated and feels automatic over time. The autonomic nervous system gives us involuntary control of the body, regulating organ systems, usually in order that we may maintain homeostasis, which is maintaining a relatively stable internal environment. The autonomic nervous system can be activated in different ways according to an individual's emotional state as well. Sensory input to the autonomic nervous system can be internal or external, and motor output is going to go to smooth muscle, cardiac muscle, and glands. There is a specialized portion of our peripheral nervous system and autonomic nervous system that deals with specific organs in the digestive system. This is the enteric nervous system, or sometimes referred to as the brain in your gut or your gut's brain. This system is going to include networks of structures that communicate with one another to help control the activity and contraction of smooth muscle that lines the digestive system, as well as glandular tissue that plays a role in the digestive system. We find the enteric nervous system located in the sheaths or coverings of tissues lining the esophagus, stomach, small intestines, and colon. This is a cross-section of the small intestines. You'll notice there is an opening on the innermost portion, which is where food passes as it's being processed for digestion. This opening is referred to as the lumen. And even though no nerves actually enter that area where digestion is going to occur, there has to be a way to monitor the conditions in the lumen throughout those areas for these structures to work efficiently. You'll notice that there is a network or plexus called the submucosal plexus. That's found here. And extends into the epithelial lining of the small intestines. The submucosal plexus contains sensory cells that are going to communicate with another network or plexus. This is the myenteric plexus. The submucosal plexus communicates with the myenteric plexus, as well as with motor fibers that can stimulate the secretion of fluids into the lumen. These fluids may contain different chemicals to aid in the chemical process of digesting food. The myenteric plexus has neurons responsible for regulating the enzyme output of adjacent organs. And these two systems, the submucosal plexus and myenteric plexus, again communicate with one another. I'm not going to ask you about the inner workings of the enteric nervous system. However, I do want you to understand that it is part of the autonomic nervous system and part of the peripheral nervous system, and it can operate independently from the CNS, the central nervous system. The nervous system includes two basic types of cells. Neurons are the most functionally important type of cell. Each neuron contains a cell body, or soma, and dendrites, which are extensions of the cell body. Both the cell body and the dendrites are designed to receive incoming information. Neurons also have a process that extends away from the cell body called an axon Sometimes you'll hear the expression axons away, because the axons carry information away from the dendrites and cell body to the axon terminal. On this slide on the left, we also see glial cells. Glial cells, or neuroglial cells, include a variety of types of cells that support the neurons and the activity of those neurons. Let's discuss neurons a little bit further. Neurons conduct nerve impulses that help us communicate information about sensations we receive, as well as produce movements, induce thoughts, and neurons have an extreme longevity. They cannot divide. They can't undergo mitosis, therefore they cannot be replaced. Neurons have a very high metabolic rate and cannot survive without oxygen. Let's take a closer look at the pieces parts of a neuron. Neurons have a cell body, which is this enlarged portion of the neuron. The cell body is often referred to as the soma. Within the soma, or cell body, we see that there is a lot of rough endoplasmic reticulum that is often referred to as a chromatophilic substance. The term chromatophilic means color-loving because when neurons are stained with color, these collections of rough endoplasmic reticulum, also known as Nissl bodies, tend to absorb a lot of the stain. The rough endoplasmic reticulum is called Nissl bodies because that is the name of the gentleman who first noticed these under the microscope when he was looking at the soma of neurons. Remember that the purpose of rough endoplasmic reticulum is to synthesize or produce proteins for use outside of the cell or for incorporation in the membrane of the cell. In the case of neurons, these proteins will act as neurotransmitters to be exported out of the cell at the axon terminal, where the secretory region exists, to secrete these neurotransmitter proteins by way of exocytosis. The cell body has smaller projections off of it known as dendrites. Both the dendrites and the membrane on the cell body serve as areas for receiving incoming information, which is why we see the fact that these are labeled as receptive regions of the neuron. The membrane on the dendrites as well as the cell body will receive incoming information. If you happen to notice the term Biosynthetic Center under the cell body, that simply means it's an area where a lot of molecules are made. We know that protein is made in the cell body for transport out of it, and the cell body also contains a lot of mitochondria because neurons require a lot of energy. They need energy not only to produce the proteins, but also to maintain an electrical potential on the membrane of the neuron, which we'll discuss soon. Extending away from the cell body is a special structure called the axon. We used to say axons away because they carry electrical impulses away from the cell body or soma towards the terminal branches. The axons are also sometimes referred to as fibers. Sometimes axons are covered by a special covering called a myelin sheath, which helps conduct nerve impulses more quickly than if there were no myelin sheath on the axon. Imagine an electrical wire having a covering of plastic to allow electricity to flow more efficiently. The myelin sheath helps do the same thing. However, you will notice that there are little gaps between the cells that make up the myelin. The cells that produce myelin in the peripheral nervous system are referred to as Schwann cells. The little gaps between the Schwann cells are myelin sheath gaps, otherwise known as nodes of Ranvier. We'll see this again, and I wanted to repeat it to you so that you understand the term better. There is also a special area of the axon where it initially forms off of the cell body. This triangular area of the axon is known as the Axon Hillock. We'll discuss that in just a second. As the axon branches, the terminal branches are sometimes referred to as telodendria, which is T-E-L-O-D-E-N-D-R-I-A. The telodendria split apart prior to the axon terminals, where we see either the axon terminal as an enlarged portion of that branch or a synaptic end bulb or synaptic end knob. Truly, the axon terminals and the synaptic end bulbs are basically the same thing. They both have a lot of mitochondria and vesicles containing neurotransmitters. Sometimes the vesicles containing neurotransmitters are referred to as synaptic vesicles. Again, axon terminals, synaptic end bulbs are used very often interchangeably. One of the things that you need to understand on this picture is that axons in the peripheral nervous system group together as fibers that form parts of a nerve in the peripheral nervous system. Where axons run together in the central nervous system, they have a different term, which is a tract. We'll see this again. Here we have another picture of a neuron. You can see the dendrites and the cell body, which are the receptive area of incoming information for the neuron. That's why those areas are referred to as the input zone. Any input coming to a neuron, either from a sensory receptor, a sensory neuron, or another neuron, is going to form a communication known as a synapse with a dendrite or the cell body of a receiving neuron. This information can get added up and spreads to the summation zone known as the Axon Hillock. Remember that Axon Hillock is the area that has a somewhat triangular appearance between the cell body and the axon. All of the incoming nerve impulses will combine at the Axon Hillock. They get added together. Now, impulses that are excitatory will lead to excitation, which is a continuation of an electrical impulse. Input that is inhibitory will actually decrease the neuron's ability to send an electrical impulse down its axon. So the information that's being received makes a difference as to whether or not electrical impulses will continue or be stopped. The axon is also referred to as a conduction zone because if a nerve impulse is spread down the axon, it is essentially conducting electricity. The axon contains many sodium and potassium voltage-gated channels. These are proteins that change shape to allow sodium to come in and potassium to go out, depending on the voltage of the membrane of the axon. The output zone is at the very end of the axon at the axon terminals or synaptic end bulbs, because that's where, if a nerve impulse is sent down the axon, more neurotransmitters will be released at the axon terminals or synaptic end knobs. These end knobs contain voltage-gated calcium channels. If you remember from our discussion with the motor end plate and the neuromuscular junction with skeletal muscle contraction, as electricity comes down a motor neuron and reaches the synaptic end knob, it opens up those calcium voltage-gated channels, which allow calcium to go in, and that causes the synaptic vesicles to fuse their membranes with the membranes of the synaptic end knobs, releasing the neurotransmitter acetylcholine to diffuse across the synaptic cleft to bind to acetylcholine receptors on the motor end plate of skeletal muscle. If we look at neurons according to their structural classification, on the far right of this picture we see multipolar neurons. Multipolar neurons have a single axon and many dendrites coming off of their cell body. So if you go to the soma or cell body and just count the processes coming off of it, you can see there are a lot. There are multiple processes coming from the cell body, therefore it is multipolar. Multipolar neurons are the most common type of neurons in the nervous system, and motor neurons are multipolar neurons. If we look at the next type of structural classification for neurons, we see on the far right of the picture bipolar neurons. These are named bipolar because if we go to the soma or cell body and count the number of processes going to or coming from the soma, we find there are 2, hence the term bipolar. These neurons are very rare. They're found in the retina, which is the nervous tissue in the back of the eyeball, as well as in the olfactory mucosa in the nose. Then finally we have unipolar. Some books call these pseudo-unipolar neurons because there's just one process coming from the cell body, and information is thought to bypass the soma or cell body and basically come in and keep going. These unipolar or pseudo-unipolar neurons are found in sensory neurons in the peripheral nervous system. You should know where these neurons are found. Next, we're going to discuss the neuroglial cells. Neuroglial cells are found in both the central nervous system and the peripheral nervous system. We're going to discuss the types of neuroglial cells found in the central nervous system first. There are 4, including star-shaped cells known as astrocytes, tiny cells known as microglial cells. Remember, the prefix micro means tiny. We also have epinole cells. You can see the term epinole cells down on the bottom left. Lastly, in the central nervous system, we have oligodendrocytes. Remember how I showed you Schwann cells wrapping around an axon earlier? Schwann cells are found in the peripheral nervous system. Oligodendrocytes are cells that produce myelin in the central nervous system. Let's take a closer look at these cells. Starting with the oligodendrocytes, as I mentioned before, they are found in the central nervous system. They hold the nerve fibers together. Anytime you hear the term nerve fibers, I want your brain to think axons. Oligodendrocytes wrap their membranes around the axons, producing a covering known as a myelin sheath. This membrane has a high concentration of phospholipids, and fat works as a great insulator. Oligodendrocytes help to myelate axons in the central nervous system. Oligodendrocytes can extend their membrane to myelinate more than one axon or nerve fiber. So remember, one cell can myelinate many axons. This is not true for Schwann cells. We'll see that when we get to those, when we look at the neuroglial cells in the peripheral nervous system. Here is an example of what the myelin sheath is. As a membrane wraps around an axon, it continues to wrap and wrap and wrap and wrap. Just as if you were to take a Band-Aid and wrap it around your finger many times, there would be many layers. And those layers of membrane that has a lot of phospholipids in the membrane are going to make a nice insulation. Again, think of insulating an electric wire. It allows electricity to flow more easily. In the myelin sheath that's created by the oligodendrocytes, there are no little gaps between the myelin. That only occurs in the peripheral nervous system. So only in the peripheral nervous system do we find nodes of Ranvier. The purpose of having a myelin sheath is that not only does it insulate the axon, it allows the speed of the action potentials, which are electrical impulses, to be transmitted faster along the membrane of the axon, as well as assists in holding neurons together. Loss of myelin, otherwise known as demyelination, can cause a loss of sensation where a person might have numbness, or a loss of motor control, which could lead to paralysis. Before we go on with neuroglial cells, let's discuss myelin a little bit further. Wherever we have myelin on axons in the central nervous system, and those axons run in the same direction, we refer to that as a myelinated tract. Myelinated tracts are found in the central nervous system. And all they are groups of myelinated axons that run in the same direction. For instance, here we have a structure in the brain that connects both sides here, forming a myelinated tract in the central nervous system. In the peripheral nervous system, axons that are myelinated can form a structure known as a myelinated nerve. Not all axons in the nervous system are myelinated. Sometimes axons are unmyelinated, and if they're unmyelinated, any structure that does not have myelin is considered to be gray matter. White if it's myelinated because it's got lots of fat in the form of phospholipids and has a whitish appearance. However, gray matter if it is unmyelinated. Cell bodies and dendrites are always unmyelinated. I'll say that again. Cell bodies and dendrites are always unmyelinated. Sometimes axons are unmyelinated. Anything that doesn't have myelin is considered gray matter. In the central nervous system, collections of gray matter, which appears brown in this picture, make up structures called a nucleus, nuclei would be plural, or a ganglion, ganglia is plural. In the peripheral nervous system, we have collections of the soma or cell bodies of unipolar neurons or pseudo-unipolar neurons. And this little cell body right here makes up a structure in the peripheral nervous system called a ganglion. So where we have collections of gray matter in the central nervous system, it can be a nucleus or a ganglion. Collections of cell bodies in the peripheral nervous system form a structure known as a ganglion only. And the only thing in the ganglion in the peripheral nervous system is going to be the cell bodies of incoming sensory neurons. Remember, incoming sensory information is afferent. Outgoing motor information is efferent. Here we see cell bodies and dendrites of motor neurons and an interneuron. Because they are cell bodies and dendrites, they are unmyelinated and they are in the gray matter of the spinal cord, which also appears brown like it did in the brain. Cell bodies and dendrites are never myelinated. Sometimes axons are unmyelinated. Anything that doesn't have myelin is gray matter. Anything that has myelin is white matter. Remember, in the central nervous system, wherever we have groups of axons running in the same physical direction, even though information may be going in either direction, we have a structure that is usually referred to as a myelinated tract. In the peripheral nervous system, when axons run in the same physical direction and are grouped together, they form a structure known as a peripheral nerve. Let's build a nerve. We're going to start with a single axon. Around the axon, you may find Schwann cells creating a myelin sheath. Remember that in between the Schwann cells will be tiny little gaps called myelin sheath gaps or nodes of Ranvier. Covering the myelin sheath on top of the axon is dense irregular connective tissue known as endoneurium. If we group axons together, we form a structure known as a fascicle. The fascicle is wrapped with perineurium, and if we take bundles of fascicles of axons, which are also sometimes referred to as nerve fibers, we create a peripheral nerve that is wrapped in dense irregular connective tissue that we call epineurium. The prefix epi means above. Epineurium is above all of these structures. You'll notice in the connective tissue of the peripheral nerve that there are blood vessels within the nerve itself. This is necessary because we know neurons are highly metabolic cells. They need a good blood supply to supply oxygen and nutrients and carry away carbon dioxide and waste products. Here's an example of a nerve versus a tract. Coming from the retina of the eyeball, where we have bipolar neurons, we see that there are structures that form the optic nerve. Once the optic nerve goes through a special crossing known as the optic chiasma, it enters into the brain, becoming the optic tract. If it's outside, it's optic nerve. Once it goes into the brain, as it's entering, it becomes the optic tract. Let's continue our journey through the central nervous system and the neuroglial cells found therein with astrocytes. Astrocytes are star-shaped cells. Astro means star, cyte means cell. Astrocytes are the largest and most numerous of all the neuroglial cells in the central nervous system, and they connect to both neurons and to capillaries. Here's the neuron. You can see the astrocyte connecting to the neuron, as well as wrapping around these tiny little blood vessels that we call capillaries. Remember, capillaries are so tiny you could fit 10 of them in one strand of hair. By connecting to both the neurons and the capillaries, astrocytes create a framework for the central nervous system, and they also act to transfer nutrients from the bloodstream to the neurons. They can transfer waste products from the neurons back to the capillaries. In doing so, they act as a go-between or intermediary, because blood is toxic to neurons. If capillaries were to break inside the central nervous system, that would kill the neurons. That's what happens when someone has a hemorrhagic stroke. the bleeding from those capillaries kills neurons, damaging an area of the brain. So nutrients are transported and waste is transported by means of the astrocytes. Because the astrocytes seal off the capillaries, they help provide a part of the blood-brain barrier. We have to have that barrier, again, because blood is toxic to neurons. The astrocytes also act to soak up or trap any excess ions or neurotransmitters, so the neurons don't fire inappropriately. They can also help repair damaged neural tissue. And even though it's not listed here, a lot of text resources state that axons play a role in helping nerve impulse conduction. This takes us to microglial cells. Microglia are tiny, hence the word microglia. The prefix micro means tiny. These are tiny phagocytic cells that are derived from monocytes. These are little phagocytic cells that eat up pathogens and waste and debris. You can see the difference in the size of these microglial cells comparing when they're at rest to when they are activated. They are small, usually stationary, however, they can sometimes migrate to eat up, again, any kind of pathogens, waste products, or debris. They are the least numerous of all the neuroglial cells found in the central nervous system. The next type of neuroglial cell we're going to discuss are the Epinole cells, and this is actually the last because there are only 4 neuroglial cells in the central nervous system. Epinole cells line fluid-filled cavities in the brain that are referred to as ventricles. There is fluid circulating inside the brain as well as in a space around the brain and the spinal cord. The Epinole cells actually communicate with blood vessels in the central nervous system. Unfortunately, they did not make a picture where we could see blood vessels in the brain or spinal cord tissue on this particular picture. Therefore, I'm going to draw a capillary. As blood flows through the capillaries, the Epinole cells will transport some of the fluid into the cell and use that fluid from the blood vessel to produce cerebrospinal fluid. Epinole cells produce cerebrospinal fluid and circulate it because they have on their apical surface cilia. Remember, cilia are much larger than microvilli, and they have arm-like extensions that move stuff across their surface. So we see this producing cerebrospinal fluid because they line cerebrospinal fluid-filled cavities. And they aid in the circulation of that fluid. When we get to the ventricles of the brain, you're going to see that there are networks of capillaries, and it's where these networks of capillaries are that there is a higher concentration of Epinole cells to produce cerebrospinal fluid as well as circulate it. Next, we're going to discuss the two different types of neuroglial cells found in the peripheral nervous system. The first type of cell we're going to discuss, which you should already be somewhat familiar with, is the Schwann cell. Schwann cells form myelin in the peripheral nervous system only. They are also sometimes referred to as neurolemocytes. That's a mouthful. Schwann cells wrap their entire self around axons, and multiple Schwann cells will wrap themselves around the length of just one axon. Whereas with oligodendrocytes, one oligodendrocyte could extend its membrane to wrap around numerous axons. One Schwann cell will wrap around an axon, and then we'll have multiple Schwann cells in the peripheral nervous system. As the Schwann cell wraps around an axon, it creates multiple layers of membrane close to the axon, forming the myelin sheath. Remember, the membranes of Schwann cells and oligodendrocytes have a high ratio of phospholipids and therefore have a high density of fat in the membrane, which makes for good insulation. As the Schwann cell wraps and wraps and wraps around the axon, the outer layer of cytoplasm that gets pushed out to the periphery is referred to as the neurolemma. And you'll notice that the neurolemma is kind of left at the periphery because there's a tight wrapping of the membrane closest to the axon. You'll also notice that the nucleus of the Schwann cell gets pushed out to the periphery as well. This forms the myelin sheath in the peripheral nervous system and is also essential for nerve regrowth. Schwann cells play a role in regenerating axons to a certain degree. When axons are damaged, there is a limited amount of regrowth that can occur with axons. The last type of neuroglial cell in the peripheral nervous system, remember there are only two in the peripheral nervous system, Schwann cells and satellite cells. If you remember those two, all the other ones belong in the central nervous system. Satellite cells are cells that cover up and support the neuron cell bodies of unipolar or pseudo-unipolar neurons in the peripheral nervous system. You can see the satellite cells covering over the cell body or soma of that unipolar neuron, which is a sensory neuron carrying incoming sensory information from the peripheral nervous system going towards the central nervous system. The satellite cells cover and support cell bodies not only for sensory neurons, but also sensory neurons that are found in the autonomic ganglia, which we'll discuss when we get to the autonomic nervous system. They function like astrocytes in and of the way that they help control and regulate the concentration of ions around the soma so that the nerve doesn't fire inappropriately. They do not play a role in the blood-brain barrier because they're located in the peripheral nervous system. They don't get close to the brain. Here we see satellite cells as well as Schwann cells in this picture on the left. And you can see the numerous Schwann cells required to produce myelin. The little gaps between the Schwann cells are called myelin sheath gaps or nodes of Ranvier. And we can also see the Schwann cells on the picture of the right. However, the ones in purple on the actual soma or cell body are the satellite cells. Since we're discussing myelin, we're going to take a look at multiple sclerosis. I'm going to disappear from your screen. Multiple sclerosis is thought to be an autoimmune disease that causes inflammation first of the myelin in the central nervous system, and then after the myelin becomes inflamed, it becomes sclerotic. Sclerosis is a condition of becoming hard or narrowed, and that basically causes the myelin to become destroyed in the central nervous system. The picture on the upper right-hand side gives you a really good idea of a normal brain that is not affected and a brain in the end stage of multiple sclerosis on the right. As the myelin is destroyed, there's more and more obvious scarring that occurs. Multiple sclerosis is multiple scars of hardening and narrowing found in the white matter, and symptoms include both deficits in the somatic and autonomic nervous systems, which cause deficits in muscle control. People with multiple sclerosis may experience things like the inability to balance themselves, or they might have the inability to focus their eyes because they can't control their extrinsic eye muscles, and so their vision may become blurred. They'll experience numbness and tingling in their extremities and become very tired. They may experience pain, muscle spasms, dizziness, headaches. They might have problems speaking or swallowing. They may become incontinent. So there are a lot of different symptoms that present with multiple sclerosis. The initial symptoms may make a person appear that he or she is drunk. And as this is a progressive disease, eventually people will end up wheelchair bound. As it affects the brain more and more, people either become very depressed or very euphoric, where they just start laughing all the time. And normally, or typically, I should say, multiple sclerosis is seen in white females who are over 40 and who are fat. That was the way that people used to remember it. White, fat, over 40. However, when I worked as a volunteer in physical rehab in New Jersey, I saw an African-American 25-year-old male who had end-stage multiple sclerosis. And I distinctly remember him riding an exercise bike in the gym, just laughing his head off. He was riding and going, ah! And he was end-stage MS, which is very sad. Unfortunately, this is a progressive disease. I know that there are multiple types of pharmaceuticals that are being investigated to slow or stop the progression, but I am not aware of anything that's really been effective in stopping the progression or even slowing the progression of multiple sclerosis. Interesting that there is a food additive known as NutraSweet or acetylfame K or aspartame. They're all the same thing. And originally, when this food additive, this artificial sweetener, came out, one of the things that people noticed was happening was that it could cause signs of dementia, disorientation, forgetfulness, signs and symptoms of multiple sclerosis. So if you know anyone who has been diagnosed with MS, it might not be a bad idea to make sure that that person understands that artificial sweeteners may contribute to the severity of the symptoms. That ends this slide presentation. We're going to go on to the next slide, I think, or is there one more? Oh yes, Guillain-Barre Syndrome. Guillain-Barre is a demyelinating disease of the peripheral nervous system. This is also considered an autoimmune reaction where sensory and motor deficits are most common, and autonomic failures can lead to changes in the rhythm of the heartbeat or drops in blood pressure, especially when a person stands up, which is known as orthostatic hypotension. Some of the risk factors may be association with immunization and is often or frequently preceded by either mild respiratory or intestinal infections. Guillain-Barre is usually self-limiting, which means that it will resolve over time, and it can vary in terms of the length of time that it is in effect. It progresses over hours to days, but some people may live with Guillain-Barre for months, maybe a few years, but it usually does tend to resolve more quickly than that. There is minimal muscle atrophy because it usually does resolve fairly quickly. It can lead to symmetrical paralysis, usually begins in the lower extremities and goes up the body bilaterally, where people notice weakness. Ataxia is the inability to walk. Bilateral paresthesia, so they're feeling both sides with numbness, tingling, progressing to that paralytic state. And when it gets to the point where they have that symmetrical paralysis, then they can experience problems with breathing, talking, swallowing, bowel and bladder function. So that is Guillain-Barre syndrome. And this is the last slide for this presentation.